Hello girls, I hope you're staying safe at home. To continue with the seeds, today we are going to discuss some experiments on germination. Okay, the first experiment is to prove that water is necessary for germination. So for this, what do we have to do? We have to take two beakers and mark them A and B. Okay, supposing you take beaker A and beaker B. In beaker A, we place some seeds of green gram, okay, or chickpeas on wet cotton wool. So here we'll take some wet cotton wool and there we will place some seeds of green gram. And in beaker B, we will place some seeds, but the cotton is dry. In A, the cotton is wet. In B, the cotton is dry. Okay, now we keep both the beakers at ordinary temperature in an ordinary room. In a few days, what will you see? The seeds in beaker A will germinate, but not in beaker B. Why? Because beaker A we had put in wet cotton, so the seeds obtained water from the cotton that was wet. But beaker B, nothing will happen. You will not see any signs of germination. Now next is an experiment to prove that suitable temperature is necessary for germination. Okay, again the same thing what we do, we take two beakers A and B, okay, and we take wet cotton on both, and then we place seeds again, dry seeds on top. Now we take beaker A and we keep it outside in an ordinary room that is normal temperature, and this B we will put it in the refrigerator. So in a few days what will you see? You will see that the seeds in beaker A will start germinating, but not in beaker B because the temperature inside the fridge is very less. Okay, so this shows that a suitable temperature is important for germination. Okay, so next we are going to learn about an experiment which proves that air or oxygen is necessary for germination. So here what do? Is we take two conical flasks, okay, so one is A and the other one is B. Now in each flask we take wet cotton wool and spread it inside. This is the wet cotton wool inside, okay, and then we place some soaked gram seeds on top of the cotton wool. Now what do? Is we take a small test tube which contains alkaline pyrogallic acid. And what is the function of alkaline pyrogallic acid? It absorbs oxygen. Okay, so by means of a thread we take the test tube and then lower it down here in in conical flask B, and in conical flask A we take plain water in a test tube and lower it down. We keep the tube hanging by fixing a cork in the mouth of the conical flask. Okay, now we take the two flasks, this experimental setup, and we keep it in an ordinary room under ordinary temperature. So after a few days, what will you see? The seeds in flask A will germinate, but the seeds in flask B will not germinate. Why? Because flask A contains oxygen, but flask B does not contain any oxygen because it has been absorbed by the alkaline pyrogallic acid. Okay, so this shows that oxygen is required for germination. Now next we move on to the 3-bean seed experiment. In the 3-bean seed experiment, what do? We take 3 air-dried bean seeds and we take a glass slide, okay, and then we tie the bean seeds at 3 positions, 1, 2 and 3, okay, and then we take a beaker which is half filled with water, you can see here, and place the glass slide in such a way that one seed is right above, it is not receiving the water. One is right at the center, half in water and half above, and the other one is dipped in water. That means it is right inside the water. It is not receiving any air or oxygen. So we leave the setup for a few days, and what will you observe after a few days? You will see that the middle seed germinates. The one at the middle will germinate. You'll see roots, you'll see the shoot here, okay? And because why? It is getting both oxygen and water. The top seed, this seed does not germinate. Why? Because it gets only oxygen, but it is not getting any water, okay? And the bottom seed, it germinates for some time, and after some time it will stop germinating. Why? Because it is getting water, but it is not getting enough oxygen. So this experiment is known as the 3-bean seed experiment, to show that water is required for germination. Then of course oxygen is also required for germination. Now moving on to the types of germination. Now both epicotyle and hypocotyle of a seed never elongate together during germination. It is either the epicotyle or hypocotyle that elongates. If you remember, children, I explained about the epicotyle and hypocotyle when we were discussing the structure of bean seed. If you remember, the hypocotyle is the region between the radicle and the point of attachment of the cotyledon, and epicotyle is the region between the point of attachment and the plumule, okay? So these two regions do not elongate at the same time. So it is either the epicotyle or the hypocotyle that elongates. So if the epicotyle elongates, the cotyledon remains underground. So epicotyle elongating means from the point of attachment, if I draw a rough diagram, let me show you, children. Yeah. So this is the epicotyle and this is the hypocotyle. So here I just told you, if the epicotyle elongates, this will become long. So what will happen? The point of attachment is here below the epicotyle. So what happens to the cotyledon? It will remain under the ground. Okay, so in this case, when the epicotyle elongates, the type of germination is hypogeel germination. Okay, but if the epicotyle elongates, if this region elongates, what will happen? It will push the entire thing above the ground. So what happens? The cotyledon will come above the soil. So this type of germination where the hypocotyle elongates is known as epigeel germination. Example of hypogeel germination is pea and gram. An example of epigeel germination is castor and bean. So here you can see the difference. In hypogeel germination, the cotyledons remain underground. Why? Because the epicotyle elongates faster, and you see it especially in monocotyledonous seed. In epigeel germination, the cotyledons are pushed above the ground because the hypocotyle is elongating, and you will see this type of germination in dicotyledonous seed. Now, a method to observe the stages of germination. What do? We take sterilized sand in a glass vessel in which filter paper, ordinary white paper, is lined besides the inner wall, and in between, what do we place? We place seeds between the paper and the glass. When water is added to the sand in the vessel, seeds will absorb the water and germinate in a few days, and you can see what type of germination is shown. Okay, now germination in some common seeds. In pea seed, you know, it is hypogeel germination. Now what happens when you soak the seeds in water? It will absorb water and it swells up. Then the testa, that is the seed coat, it will become soft and it will burst open. The radicle will come out and it will start growing in the downward direction, because if you know, remember, radicle R, it forms the root, okay, and the plumule goes in the upward direction to form the shoot of the seedling. So in the earlier stages of development, you will see that the plumule is arched inside like this. Okay, once it comes out from the soil, it will start getting straightened up. So what type of germination is this? This is showing hypogeel germination, where the cotyledon remains under the soil. Next, we move on to the bean seed, which shows epigeel germination. Now, in the same way, the seeds absorb water and it gets swollen up, and the radicle will start growing in the downward direction. And in this case, since we are talking about epigeel germination, the hypocotyle is elongated. So as a result, when the hypocotyle is elongating, this portion is elongating, what will happen? It will carry the cotyledon outside with it. Okay, so an arc is formed as it comes out, and finally it straightens up, and this is the hypocotyle, which has elongated. So the cotyledon becomes the first green leaves, and it soon falls off after the main foliage leaves grow. Next, we will talk about the germination of the maize grain, which shows hypogeel germination. The same thing, the grain, the maize grain, it imbibes water. That means it takes in water and becomes swollen up. It swells considerably. Okay, the radicle then again pierces through the protective root sheet, that is the coleorhiza, and the fruit wall, and grows downwards to form the root system. Okay, and it dies off soon. After that, new roots develop from the base of the stem. Now the plumule, it pierces through its protective sheet, that is the coleoptile, okay, and moves out in the upward direction. So the two protective sheets, coleorhiza and coleoptile, will be seen as a membranous covering on the seedling. Okay, outside a membrane is there. So actually that membrane that you see are the two protective sheets, that is coleorhiza and coleoptile. Now the cotyledon, you know, in the maize grain is known as scutellum. It absorbs food from the endosperm until it finishes off. So the hypocotyle does not elongate, and germination is hypogeel in case of maize grain. Now next we talk about viviparous germination. Viviparous means giving birth to young ones. Mammals, human beings are viviparous. Now what do you mean by viviparous germination? Mangrove plants like Rhizophora Sonoracea shows a special mode of seed germination, which is known as vivipary, in which seed germinates inside the fruit when it is still attached to the parent plants. After germination, the plant drops the seedling into the soil, which develops a root, and then it fixes itself in the soil. So as I told you, viviparous means producing live young ones. So here you can see in this diagram, okay, this is the fruit. Okay, inside the fruit only, radicle, it germinates. Okay, radicle, the hypocotyle elongates, and finally it drops down, and then a new plant is formed. That means when the fruit is attached to the plant, the seed inside it germinates. Okay, next we come to the seedling. Germination in any plant ends with the formation of the seedling. That is, you can say the end product of germination is seedling. So seedling is a stage in the growth of a plant from a seed before it becomes wholly independent of the food stored in it. That means, remember, the seedling, before it forms its first green leaves, it is taking the food from the endosperm or cotyledon. Okay, so that is the seedling, because it cannot make its own food. So once the first green leaves comes out, then photosynthesis will start taking place, and then the seedling becomes wholly independent of the food that is stored in the endosperm. So the roots of the seedling absorb water and minerals from the soil. The leaves start manufacturing food for the young plant by the process of photosynthesis, which keeps growing and becomes a mature plant, and it produces flowers and seeds in this turn. Okay, with this we come to the end of this chapter, children. So the assignment for this chapter is, children, you are going to perform an experiment at home. Okay, what you do is you do the 3 bean seed experiment. You can tie the seeds in your ruler, at 3 different position, and instead of the beaker you use a glass that you have at home. And when the experiment shows results, then please take a picture and send it to me. It may take a few days, but please do it and send it to me as soon as you see the results. Till then, children, stay safe. God bless you.